Hello, my name is Dariangeli Pacheco Cruz. I am an undergraduate student from the Pontifical Catholic University of Puerto Rico, and I'm going to be sharing my summer project in collaboration with the Michigan State University. Methyl mercury exposure downregulates GABA-A receptor subunit expression in rat forebrain. The aim of this project was to determine whether GABA-A receptors are directly affected by methyl mercury exposure. And we hypothesized that methyl mercury exposure will cause a downregulation of the GABA-A receptor subunits. For a brief introduction, I'll start talking about methyl mercury, which is the most toxic form of mercury. It's prevalent in the environment through anthropogenic and natural sources. In this image on the right, what I'm showing is the cycle of methyl mercury, where mercury originates from the burning of fossil fuels and it gets into the atmosphere or soil and then to the water sources through runoff, where it's methylated by sulfate-reducing bacteria and it deposits on plankton to then be bioaccumulated in fish. Humans get exposed to methyl mercury through the consumption of contaminated seafood. One of the biggest concerns in the field is methyl mercury toxicity because acute and chronic exposure affects the nervous system. Symptoms include sensory and motor dysfunction, impaired learning and memory, and susceptibility to epileptogenesis. One of the main targets of methyl mercury toxicity are the GABA-A receptors. GABA-A receptors are one subtype of inhibitory ion channels called gamma-aminobutyric acid, or GABA for short. GABA-A receptors have an heteropentameric structure, meaning they are composed of 5 different subunits that can arrange differently. The most common subunit arrangement we find in our nervous system is alpha 1, beta 2, gamma 2. These receptors are permeable to chloride, leading to an inhibition upon opening. They are abundantly present on postsynaptic neurons. Strong evidence shows that methyl mercury is associated with impaired function of the GABA-A receptors. Mutations in GABA-A receptors are associated with genetic epilepsies, and damage to GABA-A receptors causes hyperexcitability and increased seizure susceptibility. Most importantly, methyl mercury is associated with epileptogenesis. For the experiments, we used postnatal day 4 Sprague-Dawley male rats, and the exposure started at postnatal day 5. Exposure was for 15 or 30 days, and the third group was exposed for 30 days and left for a washout period for another 30 days, and we will call that the 60-day group. The groups were exposed to different dosages of methyl mercury, an untreated control group, a 0.75 milligrams per kilogram per day group, and a 1.5 milligram per kilogram per day. The rats in the washout period group were left untreated to see whether if the effects of methyl mercury exposure was reversible. The animals were weighted daily for dosage calculations, and at postnatal day 20, 35, or 65, the rats were euthanized and the isolation of the forebrain was performed immediately after. The tissue was preserved by bathing it in liquid nitrogen immediately after excision, and then it was stored at minus 80 degrees Celsius until further use. The selected tissue was homogenized and RNA isolation using the tri-reagent protocol was performed. The samples were nanodropped for quality check. A 1.9 to 2.0 purity ratio was accepted. Then, real-time PCR was performed to obtain cDNA. The applicants were nanodropped to check quality and concentration of the samples. A 1.8 purity ratio was accepted. qPCR was performed to quantify gene expression, and the housekeeping genes used were GABDH and beta-actin. 2 delta-delta CT analysis was performed to compare the gene expression. Here we have our results. In the y-axis, we see gene expression. The dotted line represents control gene expression, or 1.0, Expression below the dotted line represents underexpression of the gene when compared to controls, and expression above the dotted line represents overexpression of that gene. On the x-axis, we see the genes that were measured. When we compare gene expression for different days of exposure, 15 days shown in black, 30 days shown in dark gray, and 60 days shown in light gray within the same dosage, we can see significant differences in gene expression between the groups. The rats exposed for 15 days and 30 days show a downregulation of gene expression in all subunits, while the 60 days group that had the washout period showed an upregulation of the subunit gene expression. So the main takeaway of our results are that gene expression is affected, with the majority of those subunits being downregulated at the highest concentrations of methylmercury exposure. In addition, the 60-day group show an upregulation in both doses, leading to think there may be a reversible effect in the gene expression. In summary, the data shows that methylmercury has effects in the downregulation of the receptor subunits. At 30 days of exposure, 0.75 and 1.5 milligrams per kilogram per day, the alpha 1 and beta 1 and 3 subunits were downregulated. And those subunits are important because they have the GABA binding site. At 60 days of exposure, the 0.75 and 1.5 milligrams per kilogram per day, we can see an upregulation of all the subunits. So overall, methylmercury downregulates the GABA-A receptor subunits studied. Our results contribute to the understanding of methylmercury alterations and neuronal inhibition, and GABA-A receptor impairment, which we know can lead to epileptogenesis. I want to thank the PI, Dr. William Atchison, the program coordinator, Melissa Hyman. I also want to thank the best mentors, Alexandra Colon and Kimberly Rivera, for putting this whole project with us. The hard work paid off and turned out to be an amazing experience. I'm grateful that I had you guiding me and teaching me so much in a short period of time. The data I analyzed was collected by Nicole Colon Carrion, and without that, there will be no project. And to my teammates, Julian Torres and Yarimel Rosado, for helping me work out everything, including myself. Thank you for your attention, and if you have any questions or doubts, please don't hesitate on asking them.